The morphology, size, and quality of cells, starch granules, and protein bodies in kernel determine the weight and quality of seed. In this study, we present the simple preparation of whole kernel embedded in AR white resin and establish a simple dry sectioning method of whole kernels.
We choose AR white resin as embedding medium for two major reasons. First, it is low viscosity and strong permeability, leading to its good applications in preparing the smiting sections of seeds, especially for cereal mature kernels with large volume and high starch content. Second, the sample embedded in AR white resin can be stained easily with many chemical dyes to clearly exhibit its morphology of samples under light or fluorescent microscope. The prepared sections can be specifically stained to exhibit the cells, starch granules, and protein bodies clearly in any regions of whole kernel, and the morphology parameters can also be quantitatively analyzed. The method contains 4 processes. Here, we use mature maize kernels with large volume and high starch content as a sample to exhibit the processes step by step. To begin, clip kernels into a sample bottle. Add about 10 ml of 2.5% phosphate buffered glutaraldehyde, 0.1 mol, pH 7.2, and place it at 4 degrees centigrade for 48 hours to fix samples. Then take out the kernels and slice them longitudinally or transversely with 2 to 3 mm thickness using a sharp double-sided blade, and fix them for another 48 hours.